Hello, my name is Daniel Garcia, and I'm a research scientist at Moderna, and I will be speaking about the mRNA construct. The key to understanding the mRNA construct of Moderna's vaccine is to analyze the structure of the SARS-CoV-2 virus. And coronaviruses are coated with a crown-like trimeric spike glycoprotein. The full-length S protein of the SARS-CoV-2 virus consists of an S1 and an S2 subunit. The S1 subunit harbors the receptor binding domain, and the S2 subunit anchors the spike to the viral envelope and enables fusion between the viral envelope and the host cell membrane. Now that we understand the structure of the virus, we can study the fundamentals behind Moderna's vaccine. There are two categories of mRNA construct that are currently evaluated: non-replicating mRNA and self-amplifying mRNA. Both have in common a cap structure of 5' and 3' untranslated regions, an open reading frame, and a 3-poly A tail. Self-amplifying mRNA differs with the inclusion of genetic replication machinery by encoding additional replicase components able to direct intracellular mRNA amplification. In this illustration, non-replicating mRNA constructs located in the cytosol are immediately translated by ribosomes to produce the protein of interest, which undergoes subsequent post-translational modification. In contrast, self-amplifying mRNA constructs are immediately translated by ribosomes to produce the replicase machinery necessary for self-amplification of the mRNA. The self-amplified mRNA constructs are translated by the ribosomes to produce the protein of interest, which similarly undergoes subsequent post-translational modification. Finally, the expressed protein of interest are generated as secreted transmembrane or intracellular protein, and immune responses detect the protein of interest. The full-length S protein, S1, and the receptor binding domain are capable of inducing highly potent neutralizing antibodies and T-cell mediated immunity. Since Moderna's mRNA 1273 uses non-replicating mRNA construct, the vaccine mRNA construct only encodes for the pre-fusion stabilized spike glycoprotein of the SARS-CoV-2 virus. Now I will walk you through the mRNA 1273 construct. This is the entire genome of the SARS-CoV-2 virus. But as I mentioned earlier, we should only focus on the surface glycoprotein portion of the mRNA. This is the mRNA construct of Moderna's mRNA 1273. As you can see, a lot of the features that I mentioned earlier can be seen in this mRNA construct, such as the 5' cap, the 5' untranslated region, and the region that codes for the surface glycoprotein, as well as the 3' untranslated region. This is a more detailed version of the mRNA 1273. In which the amino acids are included as well.
Hi, I'm Leon, a researcher at Moderna, and today I'll be telling you about some of the theory behind our vaccine for the severe acute respiratory syndrome coronavirus 2, the virus which causes COVID-19. The mRNA of our vaccine is derived from the spike proteins on the coronavirus molecule. Once this mRNA is then derived, it is placed within a lipid nanoparticle that acts as the transfer mechanism to the cells of our body. Once this transfer is complete and the mRNA is released into our cells, the ribosomes are then used to produce two types of proteins. The first, MHC2, and the second, MHC1, can then both be expressed on the cells. MHC2 proteins are expressed only on B cells, macrophages, and dendritic cells, while MHC1 proteins are expressed on all cells within our bodies that contain a nucleus. When these cells are expressed, immune system cells are then attracted. One type, called T helper cells, are attracted to the MH2 proteins that are being expressed and interacts with those expressed proteins in two ways. First, one protein from the T helper cell interacts with the expressed protein that way, and then another protein on the T helper cell interacts with the MHC2 complex. Once these interactions are complete, the T helper cells are activated and start producing cytokines. Once these cytokines are being produced, they then interact with our B cells, which then proliferate and differentiate, resulting in the production of antibodies that work against the spike proteins on the coronavirus. This means that if the coronavirus gets into our system and our body will already have the proper antibodies ready to act against the virus and prevent any symptoms from occurring.
Hello, I'm Alan, and I'm the production engineer here at Moderna. Today, I will be introducing you to the manufacturing operations of the SARS-CoV-2 vaccine. I will walk you through the plasmid production, mRNA production, and also the storage and packaging process. Let's go.
Fermentation is an upstream biotechnology process where the goal is to create a large culture of the E. coli carrying our DNA of interest. The DNA of interest is carried in the plasmids of the E. coli, which are circular strands of DNA that also carry our DNA of interest. And the goal of fermentation is to replicate these E. coli bacteria and duplicate them so that we have a larger overall number of our DNA of interest. Once we have a large amount of E. coli, we use an enzyme called lysozyme to cleave a linkage in the peptidoglycan component of the cell wall, leading to cell death. This breaks down the cell wall and gives us access to the contents of the E. coli, but all we really want is the plasmid with our DNA of interest.
Here in the plasmid production process, we have our fermenter, and in here we have our finished fermented cell culture of E. coli with our DNA of interest. In order to obtain this DNA, we must purify it in the centrifuge.
There are a variety of ways used to get rid of unwanted cell contents. One of them is filtration, where microfiltration can get rid of 0.1 to 10 millimeter size unwanted particles. Another would be column chromatography, where our cell content is fed through a column with adsorbent material, and once it interacts with this material, the contents with weaker attractions, which is what we want, comes out of the tube, and that way the contents that we want to keep is purified.
So here we have our purified DNA product, and we must put it into the bioreactor to begin the mRNA production process through in vitro transcription. As you can see here, we have one of our biggest units. It's one of our state-of-the-art in vitro transcription units. And with that, we begin our mRNA production process.
In order to produce mRNA from the plasmid DNA, the purified plasmid product is put into a bioreactor for in vitro transcription to happen. In vitro means outside of a living organism. Inside the bioreactor, there is an RNA polymerase which moves across the sequence we are interested in order to produce a complementary strand of RNA, which has a 5' to 3' sequence. And part of this is a 5' cap and a poly A tail, which is on the 3' end. And these two components serve to keep the mRNA stable and protected throughout the processes it has to undergo later on. The mRNA product is purified through a method called the spin column in order to get rid of unwanted material like unused enzymes and also isolate the pure mRNA product. And how this works is the bulk mRNA material is put into a tiny test tube with the interior section. And this test tube is then spun rapidly and centrifuge, and it is also washed through the interior section. And after that, the pure mRNA product accumulates towards the bottom, and this red material we'll look at here. And this material is what will be used as the mRNA in the vaccines later on in production. At the end of the production process, the mRNA is mixed with 4 different lipids to form a lipid nanoparticle. Now this complete particle protects the fragile pure mRNA, and it is also able to enter through the cell membrane without being destroyed by the cell's defense mechanisms.
Once we have our mRNA product, it goes through a series of purification and packaging before it is ready to be distributed as vaccine doses. And remember, it has to be stored in -15 to -25 degrees Celsius.
Hello, my name is Patrick Oadiuna, and I'm a doctor from Moderna. I'm here to talk to you a little bit about the clinical trials that our Moderna vaccine is currently in at the moment. The Moderna vaccine has currently reached phase 3 clinical trials and has begun to be administered to emergency case groups across the world. Though it has reached this stage, it has not been approved or licensed by the FDA, but has been authorized for emergency use under an Emergency Use Authorization, or EUA, to prevent COVID-19 from spreading to individuals 18 years or older. There is no FDA-approved general vaccine of any kind to prevent COVID-19 at this moment. The EUA for the Moderna vaccine is in effect for the duration of the COVID-19 general vaccines EUA declaration, justifying the emergency use of our product, unless the declaration is terminated or the authorization is revoked sooner than we expected. Taking a closer look at the individual phases that our vaccine has gone through, we begin with phase 1. In phase 1, the study was done through an open-label dose-ranging study, including 25 micrograms, 50 microgram, 100 microgram, and 250 microgram doses that were administered to a randomized participant group of about 120 in size. This phase was focused on the safety and immunogenicity of our vaccine, and is still ongoing to today. Moving on to phase 2, this study was done in a randomized, observed, blind, placebo-controlled dose confirmation study and focused on the same safety and immunogenicity, but this time with a larger randomized participant group of 600 and included doses of about 50 micrograms and 100 micrograms only. This phase, just like every other phase, is also ongoing. Finally, we are entering into our current phase, phase 3, which was done through a randomized, stratified, observed, blind, placebo-controlled study. This included only one dose of 100 micrograms and was administered to a randomized group of 30,418. And this mostly focused on gauging the efficacy and also, of course, the safety of our vaccine. This is our most important and final phase, and we hope to be over and done with this phase as soon as possible. Taking a deeper look into phase 3, the following adverse reactions and their accompanying percentage of the test group were collected. Additionally, about 24% of participants reported having unsolicited adverse events occurred to them, including Bell's palsy and hypersensitivity, with an additional 1% of our participants experiencing serious adverse events, including face swelling and extreme nausea. As you can see, there has been some just cause to not fully approve our vaccine by the FDA. But we here at Moderna will strive for success and continue testing our vaccine until every member of our society can safely take it. With all this said, it's only a matter of time before our vaccine is approved fully by the world government, and then soon, the end of this global pandemic will actually become a reality. Hopefully, fingers crossed.